So when you look, on the one hand, you have all these symptoms happening in cancer, and on the other hand, you find out in the medical literature, fungi can cause every single one of them and do when they
Right.
invade cells. It's just the
Wow.
thing we're missing is there's them still being chronically there and them being the driver of the disease.
Hey, welcome back to Ultra Life Today. My name is Adam Payne. I'm here in the studio with my dear friend Kyle Drew. Say hi, Kyle.
Hello, hello.
Just bringing you up to speed, we have Mark Lintern, the author of The Cancer Resolution. You can find his book on Amazon, and you can get lots of information at cellsuppression.com, where we've been doing a deep dive in our first episode. You can find that on all your major podcast places. Go to our website, also find it there. So last week we talked about the cell suppression theory. We essentially threw out the somatic theory of cancer, the DNA defect theory, into the street, let it dribble down the sidewalk and go into the gutter. And we've gone into Mark's theory about cell suppression, how fungus actually influences a cancer cell into something called the Warburg effect, which ties beautifully into the metabolic approach to cancer. We also discussed how the somatic theory failed when they tried to take a cancer nucleus and put it into a healthy cell and expecting to see cancer. And guess what? That cancer nucleus turned healthy. And this exactly the opposite happened when they took a healthy nucleus and put it into a cancer cytoplasm. That nucleus turned beautifully cancer, just to follow along, showing that cancer is happening because of something going on in the cell itself. It's not being driven by these genetic defects. So Mark, welcome back to our show here. We really enjoy having you. Just again, thank you for an amazing book and for advancing what I think is the real underlying reason why cancer even exists. It's this fungal influence. I call it the farming approach to cancer.
Mm.
Fungus is farming our bodies for its own needs. All right, so let's talk about the Narunsky paper a little bit. You mentioned that it's not as familiar to you as the others, but let's go into something here. Tumor sequencing was designed to study the human DNA, right? You went into that in our earlier book, in your earlier episode, when you were talking about the impact of whole human genome sequencing and expecting to see all of these consistent defects in tumors, and they weren't consistent and they weren't patterned. But deep sequencing also captured everything else that was in the tissue, these ignored domains that they call it. In 2022, researchers examined that overlooked data and found fungal DNA in those regions, revealing that tumors are not sterile, but biologically inhabited environments. So what did the 2022 fungal biome studies confirm for you that you think can no longer be ignored?
Well, it's just, for me, it was the notion that, and this is the big thing that you just touched on there, is that tumors aren't sterile. And we've been looking at the disease through this lens, where we've not been taking into account this major feature, which is the microbiome. And we're now learning just very recently, last few decades, about the microbiome in the gut. I mean, everyone now knows about that, but we don't actually know the complexities and understand fully yet how that interacts with the body and how essential, well, we do know actually how essential it is to our health, but we didn't until recently. And a very similar pattern is occurring with tumors. Just the fact that we're not taking into account the influence of these microorganisms.
You know, the 2022 paper is not just significant from the point of view that they found fungal DNA. They studied 17,000 tumors from across the world, and they found not just fungal DNA in some of them, but they found patterns. They found that specific tumors had specific ranges of different kinds of fungus. And not only that, so this is what really cinches it for me, which is why I think a lot of scientists that were still kind of in the somatic theory got all of their panties in a wad on this one. It's because the outcomes of those patients was highly predictable based upon the kind of biome that was there, so that they were actually able to stratify it into like poor outcome, medium outcome, some survival, and more likely to survive. They could stratify outcomes predictably based upon the kind of fungal biome that was in the patient,
Really?
showing that it's not just the presence of fungus that's causing the emergence of cancer, but that the fungus is playing a heavy role outcomes of these different cancer types.
Wow. So you were commenting on that, Mark, some of the things that you've taken from that kind of research, brother.
Yeah, so I mean, one could argue that okay, so these microorganisms have colonized the tumor after the fact it's developed from genetic mutations or damaged mitochondria. You could argue that case, but what we find is that there's a microbiome in virtually all the tissue and all the tissues in the body, including the brain. So we're now learning that these pathogens are present, and we also see in early stages of cancer certain toll-like receptors are upregulated. So TLR2, for instance, that's fungal identification toll-like receptor. And many other things that occur in early stage cancer, they also occur with infection. So we've got to then begin to ask ourselves, these pathogens are present. They weren't thought to be present previously. They must be doing something. They must be controlling something, because the earlier paper that Ravid Straussman did, and I think what got him onto looking into this, was that he was trying to treat tumors with a particular chemotherapy agent, and that agent didn't work. But he knew previously in those particular cells that he was looking at, it had previously worked. And he found out later on that it was a particular bacteria present. It was breaking down the chemotherapy drug before it could get to the nucleus. Well, at first he just thought it was contamination. But then he thought, it happened a number of times. So then he thought, we'll just double check. I'll do some DNA sequencing on this. And that's when he found a bacterial microbiome, which is why he searched the bacterial microbiome first. And I think that was 2012. And then later in 2017, this culminated in looking at fungi as well. And then, so we've realized that we now have a tumor-associated microbiome. And like you say, there's distinct signatures to the extent that we can probably use the microbiome in tumors to give us some sort of diagnostic, or the microbiome essentially in the patient to give us a diagnostic tool to detect if cancer or when cancer might develop. So that's an interesting field coming out of this. But this is something key, because we know now that the microbiome in the gut can massively influence health. I mean, fungi in the gut can overgrow in there and cause disease in the lung separately. So Candida can do that. We begin to realize that when you start looking at how fungi are able to manipulate the local environment, modulate the immune system, it throws up this Pandora's box of information that's quite scary, really. I'm quite in awe of them, because they appear extremely intelligent and extremely capable of manipulating our cells. And I think it helps the fact that they're eukaryotic as well. They share homology with our cells too, which is why antifungal drugs are largely toxic, because they affect the same or similar pathways in us. But for me, it's when you look through the pathways that are upregulated or activated in cancer, they are the exact same pathways that are upregulated when you have an infection. Now, that really stood out to me. Now, when I've posed this question to a number of people, the answer I'm always getting back is, well, infection is one thing and cancer is a completely different thing, right?
Yeah.
But then the argument there is, okay, well, maybe you're looking at cancer through this lens of malfunction, which is clouding your judgment. Maybe the missing piece of the puzzle is somewhere in between, which is that the infection, you're assuming that the infection is eliminated. Well, what we're finding with Ravid Straussman's work and other colleagues is that no, these pathogens are actually present. So what happens if you have this upregulation of these exact pathways? The pathogen remains because of the damage to the tissue is too great and the immune system is too exhausted to eliminate it. It establishes itself within this tissue. The surrounding cells of the infected cell also switch to the Warburg effect. You now have, if this stays as it is for a long period of time, you now have a proliferative state. So you now have essentially a neoplasm, a tumor growing. Then who's to say that we're looking at after the event, many years after, and we're not taking into account that the pathogen might still be there, and maybe that's the missing link. Those pathways are regulated because of the pathogen. It remains, and over time, it stays in that state, and we're just seeing it from a later stage and assuming that it's the cell that's caused the problem because it's damaged.
One of the links that I like to take from this is that the byproducts of the cell suppression theory of what these cells are producing, this lactic acid and other byproducts of the aerobic glycolysis, are actually food for fungus. Lactic acid is a primary food source for fungus, as well as other things, which which would kind of lend itself to this idea that maybe fungus is functionally altering our cellular environment in order for its own needs, pointing the immune system away from being active against fungus. The inflammatory phenotype that cancer cells produce is designed to look for other pathogens, not fungus.
Well,
So go ahead.
I was going to say, the interesting thing is an inflammatory environment, a chronically inflamed environment is like rocket fuel to pathogens. It's what enables them and facilitates their infection. So a number of studies show that I think it might be Candida. They sit within the biofilm and they produce a metabolite called acetaldehyde, and that actually suppresses the ability of mitochondria to utilize oxygen. So what you get is this forced switch, you could say, over to glycolysis.
Yep.
Now that's interesting because it suggests that the fungus is terraforming the environment for it to survive long term.
Terraforming. It's farming, terraforming. It's pretty crazy.
But then the feature of that is that when you have damage and you have this switch to glycolysis, you're producing lactic acid, aren't you?
Yeah.
What fungus does is it recognizes the metabolites being produced in the local environment, and when they sense a certain level of lactic acid past a certain threshold, they then enter into stealth mode. They then start to attack. So they use it as a signal to the realizing that the tissue is damaged. It's an opportune moment to invade the cell because
There's blood in the water, and essentially.
Exactly. So,
Well,
and
it's
then,
food in the water, essentially.
well,
Yeah, so it's
yes,
it's extending hypha and starting to eat that food and creates its own information network. I think so. One thing about our biome, our gut biome, there's more DNA in our gut biome than there is in our
Bodies.
right? There's actually more cells in our gut biome than there are cells in our body. There's 13 trillion cells in the human body. There's more than 13 trillion bacteria and commensal bacteria and microbes in our gut.
Yep.
Now, one of the things that kind of the opponents of the fungal theory or the cell suppression theory is that, well, we haven't really measured a lot of fungus in the context of cancer. In other words, the quantity of fungal influence seems to be small. How does that kind of match against I know you talked a little bit about it, that maybe fungus is the initiator and not the cause, but you're essentially saying that as the cells get damaged and more lactic acid is produced, it's essentially producing a feeding frenzy for the fungus.
yeah, it is. But not only that, I mean, one of the big things I wanted to explain was again, always looking for consistency when trying to solve a complex problem like this. And you have everyone saying, oh, there's so many different causes of cancer. So you've got all different carcinogens, you've got
Yeah.
radiation, you've got asbestos. I mean, even night nurses have a high risk of cancer because they don't get enough sleep. How could all these things actually generate the consistency of the disease? And it comes back to this lactic acid that's being produced, the weakness of the immune system, iron overload that is generated through inflammation, because the body is intelligent. It knows that when you have inflammation, that there are these pesky little pathogens lying around to take advantage of the situation. And iron is also like rocket fuel to pathogens.
Pesky.
So in that process, your body restricts iron during inflammation, like acute phase inflammation. It's called anemia of inflammation. And the whole point of that is to stop the pathogens from getting access to it so that they can resolve the tissue healthily and to turn it back to homeostasis.
Yeah.
But if you have prolonged damage, it doesn't matter what carcinogen it is. It doesn't matter what form of inflammation is being caused or caused by. That generates certain conditions. And that is a switch to lactic acid, prolonged switch to producing lactic acid through glycolysis, inflammation, which opens up the pores of the cell, essentially, enables more easily infiltrated cells. Iron overload, because now the iron can only be sequestered for so long before it then becomes freely available. And that's when it becomes damaging to the cell, and it becomes like rocket fuel, like I say, more fuel, like lactic acid is fuel to the pathogen. But those two also suppress the immune cells at the site of cell damage. So what you've got is essentially this little conclave of damage that feeds the pathogen, but also suppresses the immune cells at the proximity of the cell damage. So they can't access the pathogen when it invades the cell intracellularly. So in effect, you've got this little niche that protects the pathogen, unless you provide it with some extra help. Good food, get rid of whatever toxins causing the initial chronic damage in the first place. You're going to get this process where the pathogen is triggering a response. But it's not just a response in that one cell. This is the thing. We're not seeing that fungi are present in every single cell of the tumor. They don't have to be. What you have is that the body works communally together to solve any particular problem. And you just need one cell to be infected with a pathogen, and hundreds, if not thousands of cells in the periphery will trigger the Warburg effect as well, because that whole process is an anti-infection process. You're reducing the sugar availability, the glucose availability in the surrounding tissue. You're stiffening the cell. You're generating reactive oxygen species. All these response is hostile to fungal infection or any particular type of infection. But essentially what you have is you have a load of cells that aren't infected, but they're switching to the Warburg effect in response to that one cell or a couple of cells. So it's like a hive mind in a sense. They're being programmed. So this partly explains why the immune system isn't able to attack most of the tumor, because most of the cells are classified as self. They are just doing what they should be doing in the process of an infection. So the immune system is not going to attack them. And then when you've got immunotherapy, what you're effectively trying to do is you're trying to force your cells then to attack your own cells, which is why we do get an issue with autoimmune diseases with immunotherapy.
Yeah.
So that kind of explains that. Really, it's the ability to infiltrate the cell, which is now compromised because of the excess lactic acid that feeds the tumor and feeds the pathogen itself, and the iron overload, which blocks access to the immune system, which causes this chronically inflamed environment that needs extra help, really, to resolve the problem.
That's fascinating. I got to tell you, I wanted to alert people real quick, Mark, to your website, cellsuppression.com, where I was able to get this cell suppression theory synopsis that you wrote, which is excellent. And I'm going to ask you about something. When we're talking about the way cancer cells behave and the way that fungi behave, what we see is a lot of similarity. And I wanted to read something for you because I think you might get a kick out of this. You mentioned in your book several times Dr. Milton White, and my mentor, Doug Kaufman, spent time with him before Dr. White died. And Dr. White and Dr. Elizabeth Moore Landacker began looking at some of the similarities between cancer and fungus. And it was very, very simple. Both metabolize nutrients anaerobically, both have to have sugar to survive, et cetera, et cetera. But there is a book that I have to turn you on to from 1957. Johns Hopkins put this out. And they were starting to see this. Clinical and immunologic aspects of fungus diseases. I want to read this to you. Okay. On page 11, pulmonary coccidiomycosis, a fungal lung infection. It says, quote, suggestive of metastatic malignancy. It goes on to say, you're not going to be able to tell the difference between lung cancer and a fungal lung infection. Page 115, blastomycosis, frequently mistaken for squamous cell carcinoma, Mark.
Yep.
This one's great. Page 153, disseminated histoplasmosis, a fungal infection, is found to coexist with leukemia, lymphosarcoma, sarcoidosis, Hodgkin's disease, and is much more frequently than statistically justifiable based on coincidence. In other words, they're seeing these things coexist, and what the authors were suggesting is is it cancer or is it fungus? But in 1957, they were seeing this, Mark. And fast forward to today with your excellent cell suppression theory. In your book, I know you can't see this, but you have 2 full pages, just in your synopsis paper of the ways in which cancer is characterized and the link to fungal infections, chronic inflammation being a hallmark of cancer initiation, fungi trigger and modulate inflammation, influencing infection. You start to see the similarities between cancer and fungal infection, and it's just line after line, page after page. I wonder if you could comment a little bit about finding those similarities.
Yeah, it all really came down to trying to explain the hallmarks of cancer because that's the consistency. I was in contact with Thomas Seyfried and he suggested if I am going to be creating anything like a theory of any kind, all theories pretty much are being assessed and validated by those hallmarks. So the more hallmarks you can explain, then the more accuracy the theory is likely to be. Not only explain those 10 hallmarks, but I came across through my research at least 20 other features of cancer that are related and need to be explained as well. And I explained all 20 of those. And they all relate to all these characteristics. And when you look, on the one hand, you have all these symptoms happening in cancer. And on the other hand, you find out in the medical literature, fungi can cause every single one of them and do when they
Right.
invade cells. It's just
Wow.
the thing we're missing is there's them still being chronically there and them being the driver of the disease. For instance, I mean, even people will say, well, what about mutations? Because you have a higher risk of certain mutations causing cancer, P53, CDH1, APC. You look at these and they're all related to, well, P53 modulates the immune system for one.
Yeah.
So if it's mutated, you have the reduced ability of your immune system to be alerted to the problem and various other aspects of immune functionality, which facilitates infection. CDH1, APC, one gene, the APC gene, and I think HER2 as well, they reduce the ability of your cells to activate E-cadherin, or the tight junctions that are around the surface of the cell that protect the cell from infection and keep cells gelled together. So you've got a link there between metastasis, where these tight junctions are reduced in cancer, but these genes actually reduce the E-cadherin in those cells. Now, fungi specifically reduce E-cadherin on purpose in order to invade the cell because it's a barrier to infection. So you have,
Yeah.
so not only, I mean, the BRCA gene, for instance, that produces a protein that repairs the mitochondrial DNA. So when you have compromised mitochondria are a major defense mechanism of the cell, then you have an ability of pathogens to more easily invade the cell. So it's facilitating the infection. Then you have toll-like receptors being upregulated. You have the Warburg effect. You have programmed cell death. So pathogens, intracellular pathogens, want to survive intracellularly. And the programmed cell death mechanism is a way of a last resort, really, of killing the pathogen.
Mm.
So the pathogen blocks that process. So they invade through particular
Do you have some?
receptors
I
that
have one.
are growth receptors. So they trigger cell growth as well.
Yeah.
And many more.
Mark, real quickly, I know that we're drawing down to the end of this particular episode, but could you just touch on real quickly, what do you feel the role of mycotoxins are,
Ooh.
or fungal poisons? What are your thoughts on mycotoxins and their influence or presence in cancer?
Well, they're going to work primarily as a carcinogen, or at least as a carcinogen, in the sense of what carcinogens generally do: cause mass inflammation, damage to cells, weaken the immune system. But these metabolites, they have many other different functions. So another one which is produced by fungi and bacteria as well, but is nagalase.
Mm.
So that actually breaks down macrophage activation factor receptor on the cell, which prevents macrophages being alerted to the presence of the fungus. So it's
Yeah.
a suppression mechanism. So all these metabolites do various different things, like acetaldehyde I mentioned earlier. It
Yeah.
prevents mitochondria from using oxygen, so forcing this shift, which then benefits the pathogen because they feed on the lactic acid and the inflammation that occurs. So mycotoxins are, I think, extremely important to remove from the body if you can, as a patient, and they may not necessarily just be being caused or developed or processed by the microbiome in the body or the fungal pathogens in the body. You need to be checking your food and the environment around you, but they definitely won't help with the situation. They're going to be helping the fungal pathogens with the invasion process, basically, by generating more inflammation.
Very good.
Well, let's wrap up this segment
Yep.
with Mark Lintern, and the author of the Cancer Resolution, also known as the author of cellsuppression.com, with this wonderful synopsis paper that you can grab there
Yep.
and learn so much more. You can listen to all of the episodes on any of your favorite podcasts. I'm Adam Payne here with Kyle Drew, and this is Ultra Life Today. So come back next week for the third episode with Mark Lintern.